Across the living world, green plants save sunlight inside quiet grains of starch. In wheat, barley, rice, and maize, this reserve fills the sleeping seed. In potatoes and sweet potatoes, starch gathers safely beneath the dark soil. Each granule is a packed library of glucose, folded tightly into pale layers. For a hungry embryo, that library is full of food, but locked. The chains are too large to cross membranes or feed young cells directly. Water can soften them, and warmth can make the granules swell. But unlocking starch cleanly requires a smaller, wiser, living kind of touch. Long before laboratories, germinating seeds already carried the answer within. As a barley grain wakes, beta-amylase waits inside its starchy tissues. It helps turn stored starch into maltose, a portable sugar for growth. That sweetness feeds new roots before green leaves can harvest their own light. Sweet potato, soybean, and many grasses carry similar quiet enzymatic tools. Some bacteria learned related tricks, finding starch wherever life drops its crumbs. Human beings noticed this sweetness centuries before anyone understood enzymes at all. Malt makers spread soaked grain, then watched the kernels begin to sprout. Brewers crushed that malt with warm water, making cloudy starch turn sweet. The transformation seemed almost alive, though no yeast had begun fermenting. In 1833, two French chemists followed that mystery into sprouted barley. Anselme Payen and Jean-François Persoz extracted a powerful substance from malt. They named it diastase, from a Greek word meaning separation. Their malt extract changed starch paste into sugar without being consumed itself. It became one of the first clear glimpses of enzymatic life. The 19th century slowly learned that diastase was not one single voice. Malt contained several helpers, each cutting starch in a different way. Brewing science became a meeting place for chemistry, agriculture, and human hunger. The decisive distinction arrived in the early 20th century, after long comparison. Richard Kuhn and other chemists compared the sugars released by different amylases. They separated the end-clipping enzyme from its more random, chain-breaking companion. The name beta-amylase came from the beta form of maltose first produced. Alpha-amylase broke chains internally. Beta-amylase worked patiently from the open ends. That simple difference reshaped malting, brewing, and the chemistry of starch. Researchers then found beta-amylase in barley, wheat, sweet potato, and soybeans. Its history widened from the brewery floor to the hidden plant cell. Under a microscope, starch granules look like small moons of stored energy. Beta-amylase approaches a chain only where an outer tip is free. It grips that tip gently, like fingers finding a loose thread. Then it releases maltose, two joined sugar rings, again and again. The enzyme itself remains ready, moving on to the next exposed ending. When a branch appears, beta-amylase stops, unable to pass the fork. Other enzymes can open those branches, completing the shared work of digestion. Together, they turn rigid plant storage into flowing sweetness and usable energy. In brewing, beta-amylase helps shape the maltose that yeast will drink. Its quiet labor influences body, dryness, and the character of beer. In distilling, the same sweetness begins another journey towards spirit. In bread dough, maltose feeds yeast and helps deepen crust color. A loaf's warm aroma owes something to these hidden enzymatic cuts. In malted drinks and cereals, it helps create gentle, natural sweetness. In starch processing, beta-amylase supports syrups rich in clean maltose flavor. Those syrups appear in confectionery, baking, beverages, and fermentation feedstocks. Because the enzyme is selective, it offers sweetness with fewer harsh chemical steps. It works in water, on plant material, under comparatively mild conditions. That is why starch factories study enzymes as partners, not just tools. A biological cut can replace some heat, acid, and unnecessary waste. For farmers, beta-amylase is also a seed's private inheritance passed through harvests. Barley breeders watch it because malt quality begins inside each grain. Plant biologists follow it through seasons, dormancy, cold, and sprouting. In leaves, related starch breakdown helps plants endure the long night. The enzyme links breakfast bread to moonlit metabolism in living fields. It links a 19th century flask to modern green manufacturing and careful biochemistry. It also links old farmhouse malt floors to stainless vessels today. In animal nutrition, starch conversion can make plant energy easier to use. In fermentation, maltose can feed microbes that make foods and materials. Its story is not loud, it is a patient subtraction. 2 sugars at a time, starch becomes movement, flavor, and possibility. In every sprout, beta-amylase still performs its ancient opening beneath the husk. In every mash tun, that opening becomes part of human culture. In future biorefineries, such enzymes may help plants replace fossil chemistry. They offer industry a lesson from seeds: work precisely and waste less. The old sweetness of germination points toward a cleaner industrial future. Enzymes Bio began with a simple frustration we kept hearing. Sourcing many enzymes meant many suppliers and slow paperwork. We made it one place. Cleaner chemistry, ready for the future. Discover it at Enzymes Bio.